How do cells reproduce? Mitosis, meiosis, and the cell cycle. The cell cycle is broken down into three main parts: interphase, the longest, mitosis, the shortest, and cytokinesis, which occurs during mitosis. Interphase is further broken down into three sections: G1, the initial growth, S, the replication of genetic material, and G2, which is when the cell preps for division by growing more. Cytokinesis is when the cell divides into two separate cells, and it occurs during mitosis. Mitosis can be broken down into 6 different stages. First is interphase. Here, genetic material is messy and unorganized in a form called chromatin, which is contained in the nucleus. The cell preps for division in G1, S, and G2 phases of the cell cycle. In prophase, genetic material organizes into chromatids, centrosomes move to the poles, and mitotic spindles begin extending. In prometaphase, the nuclear envelope dissolves, chromosomes move towards the equator of the cell, and mitotic spindles continue to extend. In metaphase, the chromosomes line up at the center of the cell. Mitotic spindles attach to the kinetochores of each sister chromatid, and the chromosomes prepare to separate. In anaphase, the sister chromatids separate because the mitotic spindles pull the sister chromatids apart. Half the genetic material goes to one pole and half to the other. In telophase and cytokinesis, microfilaments create a cleavage furrow in the center of the cell and pinch the cell into two separate, genetically identical cells. Nuclear envelopes form, as do new nucleoli. Here is a review of the importance and the products of mitosis. Interphase of meiosis is just like interphase of mitosis. The genetic material is organized similarly, and the cell undergoes G1, S, and G2 phases of the cell cycle. In prophase 1, the mitotic spindles begin to extend. Centrosomes move to the poles. The nuclear envelope dissolves, and synapsis occurs when homologous chromosomes pair up and switch segments of DNA. In metaphase 1, independent assortment occurs when homologous chromosomes randomly line up along the center of the cell. Like synapsis, this increases genetic diversity in the resulting gametes after the cell divides. In anaphase 1, homologous chromosomes separate. The mitotic spindles attach to the kinetochores of each chromosome and slowly pull them apart to the separate poles. In telophase 1, the first cell division occurs. The resulting cells are diploid cells, meaning they have the same amount of chromosomes as the parent, but they are genetically unique and are not identical to the parent cell. Additionally, nuclear envelopes reform. Here is a review of the importance and results of meiosis 1. In prophase 2, the nuclear envelope dissolves, centrosomes move to the poles, and mitotic spindles begin extending. Here, the genetic material is already organized into chromosomes. In metaphase 2, chromosomes line up in the center of the cell and prepare to be divided. Mitotic spindles attach to the chromosomes at the kinetochore. In anaphase 2, mitotic spindles pull sister chromatids apart, separating the centromeres of each chromosome. The cell divides in telophase 2, resulting in 4 haploid cells. So, the 2 cells that were reduced in meiosis 1 become 4 cells after meiosis 2. These cells are genetically unique. Here's a summary of meiosis 2, including its importance and its results. For a quick review, here's the difference between meiosis 1 and 2. And finally, here is a review of meiosis versus mitosis.